Greetings from Nairobi, Kenya. This is Enabling Science. I'm your host, Bernice. Today we'll be talking about Bioinformatic BLAST, and we'll be working through some practical exercises. The cassava plant is an important crop not just here in Kenya, but across all other African nations, Southeast Asia and South America. It's a staple food for many groups, an estimated 500 million people. Despite its prevalence, the crop can still be improved. Drought, floods, and diseases can put cassava crop harvests at risk. This disrupts people's livelihoods and food security. And this is where genetics comes in. Talented scientists, crop breeders, and farmers come together to improve the traits of crops like cassava. They do this by breeding for useful genes. To do this, we often need bioinformatics. This is where we use computational technology to understand and analyze genetic sequences. One of the most widely used techniques is called sequence alignment. Let us learn more. Sequence alignment is an incredibly useful and revolutionary bioinformatic tool. It's also widely accessible. The most popular application is called BLAST. BLAST stands for Basic Local Alignment Search Tool. This will be the tool we'll be learning about today. We can carry out genetic sequencing of many organisms. This includes animals, fungi, bacteria, and also plants and crops. This will tell us the genetic code that makes up the organism. With this genetic code, we can compare or align it with other organisms. This allows us to see organisms which share similar codes. This is known as sequence conservation. We've just spoken about genetic information, that being nucleotide-based DNA or RNA. These nucleotide sequences encode for proteins or peptides. BLAST can also compare protein sequences too. It's a very versatile tool. BLAST often serves as the first step for scientists as they begin experimental work. We can use BLAST to find related genes or proteins. These are known as homologs. Identification of these proteins can prompt further biomolecular investigations. We can use BLAST to understand more about an organism's genetic evolution. We can use it to design oligonucleotide primers for polymerase chain reaction. And we can use it to see if molecular cloning and sequencing has been successful in the lab. Now let's get some hands-on practice. Feel free to pause the video so you can follow along at home. First, we shall head to the following website. There is a clickable link in the video description. We shall first navigate to this button that says Nucleotide BLAST. We want to align a gene called GBSS2. This is from Manihot esculenta, the Latin scientific name for cassava. This gene codes for starch synthesis enzyme. This could be an interesting gene for scientists to work with. We can use it to improve the nutrient profile of the cassava. BLAST accepts searches using gene IDs or FASTA sequences. These are text-based representations of peptides or nucleotide sequences. Here we will input the following gene accession number. This corresponds to a much longer nucleotide sequence. We can use accession numbers found on various online databases rather than typing out the code each time. We shall leave the default settings for now. By altering these, you can change the search parameters and stringency. So, a little on the terminology involved with BLAST. A sequence that we input to search is called a query. The sequence of that gene is aligned against an incredibly large database of genomes. The subject sequences are all the matches found from the search. There are a variety of flavors of BLAST. BLAST N and BLAST P search for nucleotides and proteins respectively. In this search, we are using BLAST N. BLAST X is another search type. This will translate products of a nucleotide sequence against a protein database. Related to this is T-BLAST X. This search helps to find distant nucleotide sequence relationships. It does this by translating an input nucleotide sequence at all 6 possible different reading frames. For those unfamiliar with molecular biology, this means all the different starting points for translation into proteins. T-BLAST-N is a search setting that does the opposite. It compares a protein sequence against all 6 reading frames in a nucleotide database. 2 other useful search types are MegaBLAST and PSI-BLAST. MegaBLAST can compare a large number of sequences at once. PSI-BLAST is a more sensitive method of finding distantly related proteins. Let us click the BLAST button here to begin the search. Hits are recorded by first finding areas of 100% sequence similarity. These are then flanked to areas of good similarity. Base mismatches and gaps are allowed. This leads to a lower score, which we shall get into in a bit. Here we are greeted with the results of our search. Here we have lots of information. I will now draw your attention to 2 of the most important aspects. First, we have this list of similar proteins. BLAST has aligned our gene sequence against other sequences to find ones with the highest similarity. For example, this predicted protein here has a query cover of 63%. Secondly, we can explore this in more detail. We can click this to see the sequence alignment itself. Here we can see which nucleotides have been conserved. We can also see which ones are different. The top line is our search sequence. The bottom is our predicted protein. Those DNA letters linked with a line are conserved. Those without a line are different. We can download or copy this sequence alignment. This can be used for further bioinformatic analysis described later in the course. You can also click the download button at the top of the page. This will give you a full tabular version of all your results. You can then use this for coding scripts of your own. There's a lot of information on this page, so take your time. Play around with the different settings. Explore all the links. What kind of data can you find? Here are some useful bits of information that I'll go through with you right now. Here are some values given for each of our identified proteins. The max score represents the score of the single best aligned sequence. Ultimately, the higher the score, the better the alignment. The total score is the sum of all the sequences that were aligned. The same goes with this value. The higher, the better. Multiple non-overlapping hits can be found within a single subject. This increases the score. Query cover represents what percentage of your input query sequence overlaps with the aligned sequence. You can have a sequence of a few base pairs, but because it fully fits to the sequence it's being aligned to, it will get 100% query coverage. Similarly, a longer fragment, which has some of its code outside the aligned sequence, will get a lower query coverage. The value here is known as the E-value or the expectation value. A lower E-value will result in a more significant alignment score. The E-value represents different alignments equivalent to or better than what can be expected purely by chance. Ident stands for identity. This is often expressed as a percentage. This is the proportion of amino acids that are identical at the same position across sequences compared. And lastly, this is the accession number. This is a unique identifier given to a particular sequence. The inner workings of BLAST could be its own entire series. It's very complex. For those interested, we have a few more resources available from the following links. These are accessible from the video description. Now, a small quiz to test your knowledge. Can you answer the following question? What type of sequences can BLAST search for? Did you remember correctly? BLAST can search for nucleotides and proteins.
Tools like BLAST are uniquely specialized, while standard search engines do not handle genetic sequences well. BLAST can insert spaces when searching to find partial matches of sequences where only some of the genetic code is the same. BLAST also just searches databases of genetic and protein sequences. Google searches for all other things too, and so is not an efficient way to search.
So now it's over to you. A chance to get a real experience with BLAST. I want you to search and compare a protein from maize. This is another important crop, especially here in Africa. ID1 is a zinc finger protein. This protein regulates flowering. Using the following FASTA formatted protein sequence, located in the video description, can you find the following? What are the 3 closest related proteins? What protein has a query cover of 83%? Take a look at the genetic distance tree. What group does your query belong to? What happens if you change to the Quick BLAST P algorithm on the original search screen? Do you see different results? Why would it be beneficial for scientists to research this protein? What are the possible improvements for the maize? Can you try to do a BLAST search for the gene ABE7 from onion, allium, cepa? How would you get the FASTA sequence? Here is a hint. Try searching for the following on a regular search engine like Google first. Post your findings in the comments. It has been my pleasure to teach you all today. Subscribe to this channel to be informed of all the latest uploads to this series. If you found this video useful, please do give a thumbs up. Until next time, see you soon.